create a functional T cell receptor. Once we have succeeded in creating a population on a daily basis as T cells develop, that's not right. As a T cell successfully completes its development, it then, of course, leaves the thymus and enters the circulation, where, like B cells, it spends most of its time going from lymph node to lymph node, sniffing out signs of infection. Just as is the case with B cells, if we create a T cell that recognizes a self protein, that would be an anti-self T cell, that T cell will either die on the spot or become paralyzed and later on be liver. There's a second aspect of the development of T cells, however, that we'll be discussing. In the case of antibodies, the tip of the arms have to just be able to tightly bind some kind of antigenic amino acid sequence typically, and we're good to go. In the case of the T cell receptor, we've got to be able to recognize the foreign peptide, but we've also got to be able to bind the presenting MHC molecule. So the T cell receptor has got to bind both amino acid sequences on the MHC molecule, any one of the 12 possible versions of class 1 or class 2 MHC, depending on and then having, and that's called positive selection. So if we create a T cell receptor, if it's not sticky enough to the MHC molecules that your body makes, it'll be rejected. Okay. If it is, it will be permitted to go on to the next day, where we make sure, and we find out, well, okay, so it can recognize the MHC molecule, but is it recognizing self peptides? That's negative selection. So it'll go through two screening processes, positive and negative selection, which we'll be getting into as we proceed to this lecture. And your book then gives you a picture of what's called one of the lobules in a thymus gland. There's lots of little lobules, and then it's in these little globules that the action is occurring. So the stem cells that arrive originally from bone marrow settle in here, and they then begin the process of development. Okay. So the process starts up here, and as we go through the stages of creating a T cell receptor, and then positive selection, and then negative selection, and then when all that is completed, it enters the circulation down here. So we have the cortical region here, and the medulla of the upper capsule. And here we have all the immature T cells called immature thymocytes, we would say. And then down here we have the mature thymocytes, which are ready then pretty shortly to enter the circulation. Here we're undergoing a positive selection, and down here we're undergoing negative selection. And it's a pretty brutal process, and more brutal even, I think, than in the case of B cell selection, where it's just essentially negative selection against anti-self. As I've said before, whereas in the case of B cells, you're constantly producing them every day of your life. You're making new B cells with newly generated antibodies of whatever random specificity that can be created. And there's a constant turnover of B cells in your body. In the case of T cells, you start making them, and once they're released, they don't die. They stay in the circulation, and over time, you build up a sufficient number of them, and once you've made, by the time you're your age now, you've made 80% or so, 90% of the T cells you're ever going to make it. You'll continue making more as time goes on. By the time you get to my age, as I've said, you're not making too many T cells anymore. And I've got all the T cells pretty much that I ever made. Now we're going to do a little sort of sidebar here. The maturation of T cells, that, as I said, occurs in distinct stages. And each of these stages is characterized by the expression of particular cell surface proteins. And as immunologists decades ago began to work out this whole process at the sort of molecular cell biology level here, they used antibodies against these cell surface proteins and a machine called a fluorescence activated cell sorting machine, basically here, to be able to pull out these one by one these precursors and characterize them, find out what's going on at the level of the gene segments which we're assembling to make a T cell receptor, and so forth. We're going to come back to this in more detail here. This shows, remember I told you that most T cells express a T cell receptor with the two chains called alpha and beta. Okay. But there's a subset of T cells, a relatively small fraction, which are express a more primitive T cell receptor called the gamma delta T cell receptor. And in the thymus, you're doing the rearrangements to make gamma delta chains, as well as at the same time you're doing the arrangements to make alpha beta chains. We'll discuss this in more detail a little later. But I want to talk, as I said, as a sidebar here, I want to talk about the fact that I want to talk about fluorescence activated cell sorting. I think many of you are familiar with it already, but it won't hurt to have your memory refreshed. Monoclonal antibody technology, by the way, was developed kind of contemporaneously with a lot of this, and it turned out to be really invaluable in being able to pull out all these different populations of cells at various stages of development. So I said here that the various T cell precursors were identified and studied by fluorescence activated cell sorting, FACS. Okay. Now, in FACS, in cell sorting, we, using laser lights and fluorescent antibodies, were able to identify cell by cell, a population of cells, cell by cell, and recognize that cell as expressing CD4 cell surface or CD8 on cell surface or whatever. And then all this, that electronics feeds here, we can then kind of tag the cell based on its fluorescence. And then we can send some cells off into one tube, maybe the CD4 cells. Some other cells present in the population, which are CD8, will be sent to another tube for collection here. And then cells with either CD4 or CD8, let's say, will go to maybe the waste flow here or some other tube. So we're actually cell sorting here, and then having sorted the cells we had in the different test tubes, and we could go ahead and proceed to characterize them. Sometimes you don't necessarily really want to collect the separated cells and do things with them. You just want to count the numbers of different kinds of cells. And we've done here, we use the term flow cytometry. Cytometry means cell measuring, of course. Okay. So there's a slight difference in terminology here between cell sorting versus flow cytometry. Often you do both, of course. You could be nothing to stop you from measuring sort of numbers of cells at the same time as you're actually going to save them to do things with them. So you often have data which has to be represented on a sort of a three-dimensional plot here. You have some cells which are going to have high or low. All cells will have high or low fluorescence, let's say. Some of them will be have, for example, certain degrees of intracellular granularity, high or low, depending on intracellular granules and so forth. Others will be big or small. As the cells pass through the laser light here, one by one, the laser shines a light through them. If the light is depending on how much light just passes right through, it's a measure of essentially the size of the cell. Okay. If it's scattered off to the side here, it's a measure of how many internal granules there are here. And so the light that passes through is captured and measured. That's a measure of the cell size. Light which is scattered, it's a mirror here. It's what we call a half-silvered mirror. Not fully silvered or perfectly reflecting, but half-silvered. So about half the light is reflected as a mirror would. The other half of the light just passes right through the prism, the glass. Okay. So the light that was scattered here is about this beam splitter, as it's called here, and then be reflected by the mirrored portion, and the photomultiplier tubes here essentially measure the photons and generate a current. Okay. So we measure the amount of transmission here with this photomultiplier tube. This light is scattered inside of each individual cell is measured by this photomultiplier tube. Okay. And then this light proceeds on here, and then if these cells had been tagged with red fluorescent antibodies or green fluorescent antibodies, maybe we had an antibody that sees the CD4 protein, and we attached a colored fluorescent tag onto that anti-CD4 antibody, maybe a red colored tag. Antibodies that recognize the CD8 protein would have then maybe a green fluorescent tag on them. Okay. So here we can now hit another beam splitter here, half silver mirror. The reflected light here is directed to a photomultiplier tube that will measure green wavelength fluorescence, and what passes through will be measured as the red fluorescence with this photomultiplier tube. And as this is happening, as we're measuring it, we're talking to the electronics here, and we can then be putting little positive. Each cell is inside a little liquid droplet of medium, basically, and you could put the machinery can put a positive or negative charge on each little droplet, in which there's on average a single cell. Some droplets have 0 cells, some droplets maybe have 2, but most of the droplets have just 1 cell. So, and then you have here's where we put the charge on here, and then we have a positively charged electrode here that the negatively charged droplets move towards, and here we have this is a negatively charged electrode here that will attract the positively charged droplets, and here we have, of course, a positive electrode that will attract the negatively charged droplets, and in this way we separate the populations of cells from each other. Okay, so you'll have some cells that have the amount of fluorescence or a measure of the number of the population will be on the y-axis here. On the x-axis here would be, I don't know, granularity or size, I guess, or right over here could be granularity. We'd also have red fluorescence or green fluorescence and so forth. So I want to show you what some of this self-sorting data looks like. First, let's so here are a population of cells. Let's say some of them are in this picture colored green. That's because they would have, let's say, an antibody against CD4 protein with a green fluorescent tag on it. Okay? Other cells are expressing CD8, and they have a red fluorescent tag antibody against CD8. Blue or whatever. Okay. By the way, in the original stuff, we had just red or green fluorescence. Now we have a whole palette of all kinds of colors that you can really tag cells with, red, green, blue, all the colors of the rainbow. Each different cell having its own unique color based on its fluorescently tagged antibody, for example. And again, here another representation. The cells, little droplets here containing what's called sheath fluid here. They pass down this narrow diameter capillary, basically. And the laser light excites them one way or another. We see how much light passes through directly. That's forward scatter. That's a measure of size. Light that is scattered off to the side is a measure of the granularity of the cells. And then we can also then measure the red or green fluorescence or whatever other colors we have in our palette, as I said. And now that, since the data that you get is three-dimensional, I showed you a moment ago an actual sort of three-dimensional drawing representing what the data could look like. But often you don't present the data as a three-dimensional cube-like representation like this. For the x and y axis, you'll have the usual x and y axes here. This would be, for example, red fluorescence on the y axis, and the intensity of fluorescence here on green fluorescence, let's say, the CD8 protein here. Okay, and then the number of cells with that intensity of fluorescence would be actually a little mountain peak coming up at you from the surface of the page here. But to represent that three-dimensional mountain peak coming out at you, you can do essentially concentric circles. Have any of you gone hiking in the backwoods and you use Geological Services maps where you can essentially topological maps? Raise your hand. I'm not talking about that topological map, right? So you could have a topological map representation here where if it's a gentle little slope, there's wide spacing between the different circles indicating a certain altitude, okay? If it's a sharp peak, then the circles of increasing higher altitude are very closely spaced and so forth. So it's intuitive. You can read here, this is a gentle little peak here, and this is a little sharp peak coming out at you. Right? So this is a whole lot of cells. This is just a few cells. A few cells and a whole lot of cells. You can do something like a dot plot, where the intensity of the number of dots is a measure of the population of that particular type of cell and so forth. You can also do pseudo color. This is like, when we get these storm fronts that go through, the weather guys on Channel 8 and Channel 11, they go nuts. They have all these graphics, and you see all these color-coded intensities and so forth. In general, of course, red is hot, high intensity, blue is cool. So you're looking. So here's a little mountain peak here. And here in red is the peak of the mountain, and then the grade down here is a little blue foothills, so to speak, of the mountain. So this is simply 4 different ways of representing the three-dimensional data that we're looking at here. Now, you see these things in journal articles all the time where they're doing self-sorting experiments. There's one point I want to make here is that the x and y axes are, generally speaking, going to be logarithmic axes. These are we're looking at the exponential increases here. So this is 0.1, 1, 10, 100, 1,000 here. So these are cells here which light up with green colored anti-CD8 antibody. Okay, so they have a lot of green fluorescence here, somewhere between 10 and 100 value for fluorescent intensity. Okay. So these cells are CD8 positive. Okay? But if we look on the axis here for the value of red fluorescence, which would be in this case a marker for CD4 protein expressed on the cell surface, so we have a red tag anti-CD4. These cells here, their value for red fluorescence is very low here. Okay? But notice it's not, it looks like it actually has a value of, 0.5 or 1 or something like that. Okay? You might say it's just low CD4. So these cells here, they are high on this axis, low on this axis. So we would say these are CD8 positive. And we would call these cells actually CD4 negative. These are cells that are actually not expressing the CD4 protein. They do have some red fluorescence, some measurable red fluorescence, but that's just background fluorescence. Okay? Some of the red fluorescent antibodies are going to stick non-specifically. They're really going to bind tight to CD4 protein if it's expressed in the cell surface. But there'll be a little sort of stickiness of the antibody to the surface of the CD8 positive cells, even though there's no CD4 protein there. So they have a low but measurable value of CD4 fluorescence. Okay? We nonetheless call these cells CD4 minus. These are cells right here which are low for CD8 and low for CD4. We would say these are CD4 minus / CD8 minus. These are what we would call double negative cells. Now they have measurable CD8 and measurable CD4 fluorescence, but that says background, we say it's negative. Okay? It's kind of important to understand how people get kind of confused about that. Okay? Now here are cells up here. Well, they are very high on the y-axis here. They have lots of CD4 protein on their surface, but they're very low on the CD8 axis here. Not zero, but we're going to say it's zero for all practical purposes. So these are cells which are CD4 positive and CD8 negative. So these cells here, CD4 positive, CD8 negative. So these are cells that we will call these CD4 T cells, and we will call these CD8 T cells. Now, in the thymus, we will find a population of cells which are double positives. These cells express both the CD4 protein and the CD8 protein. Here they are. There are not many of them, but there they are in a particular population. If we took cells from a certain area in the thymus, we would find lots of double positive cells. Most of the cells would be double positive in certain regions of the thymus. And down here, of course, double negative. Now, there will be a little two-pointer question on the upcoming second exam where I'm going to essentially show you one of these squares here. It doesn't really matter. Well, I'll pick one, and you're going to have to identify which are the cells which are CD8 positive, CD4 negative, or maybe I'll ask you which are the cells which are double positive, or which are the cells which are CD4 positive, CD8 negative. It's very simple, but we'll have to be able to recognize what we're talking about. Speaking of which, I think you all know that we'll do this afternoon's lecture topic, and then I hope that's going to leave me 45 minutes or so to go through another exam review like we did in the first exam. So if you have a friend who's going to blow it off this afternoon, you might want to, if you're feeling kindly disposed towards that friend, send them a text saying, you might want to get here. Let's see, what did I just do? Okay, so that's done. Now we're finished with that little sidebar topic of fluorescence activated T cell sorting. So let's go back to our main topic here, which is the details of T cell development, which has a lot to do with, just like for B cells, cutting and joining DNA pieces together to create an actual functional contiguous set of sequences of which function as a gene or a CD4 protein. So here, I'm just reminding you what I said to you just a moment ago in your lecture here. that the T cell receptor has two chains. It's a heterodimer. Okay, we have a gene coded by a protein coded by a particular gene here that makes the alpha chain protein. And another gene will encode a protein called the beta chain. These two proteins are have amino acid homologies, but remember they're basically more or less equivalent to the heavy chain on the arm of an antibody and the light chain on the arm of an antibody. They're evolutionarily derived. So the beta chain resembles that part of one of the arms that has an antibody. This yellow one would be part of the arm, and it would continue on down to form part of the tail, right? And then the light chain on the arm would be just standing by itself, and so the alpha chain of a T cell receptor resembles the light chain of an antibody protein. And as I'm going to tell you in a moment, you may or may not remember already, but when we talked about B cell development, we said that the early cells that are going to become a B cell, the first thing that they start working on is to assemble the boxes together to form a heavy chain for the antibody protein. If they succeed in making a heavy chain for the antibody protein, they'll proceed then to work on cutting and joining the pieces of DNA that are going to make a light chain of the antibody. And it's going to be the same deal with the T cell receptor. In T cell development, we're first going to go to work trying to bring together the pieces of DNA that are going to code for the beta chain of the T cell receptor. And then if that's successful, we'll go to work trying to bring together the DNA segments that will code, that will make a gene for the alpha chain of the T cell receptor. So it's all very analogous to what happened with the B cell. Yes?
Are those on different chromosomes or same?
Yeah, they are. I think they're on different chromosomes. So are heavy and light chain, of course, on B cell. And then, of course, there's also the thing that, as I just said a moment ago, 95% or maybe 99, I don't know what the numbers are, some very large, most T cells by far make an alpha beta T cell receptor. But there are a population of primitive T cells that make this more primitive gamma delta T cell receptor. Whereas again, for gamma deltas, delta corresponds to beta, which corresponds to the heavy chain of an antibody, and gamma corresponds to alpha, which corresponds to the light chain. Okay. By the time I got through, actually I guess I was a chemistry major in college, but by the time I got through my maths and chemistry, there's so much Greek alphabet stuff in mathematics that I pretty much knew the Greek alphabet. In my junior year abroad, I traveled in Greece, and I could pretty much read the signs and then sort of sound out. And yet sometimes I can kind of figure out like homologies in the English language have a lot of Greek and Latin roots, and it's very helpful actually. And so I don't know how much math you guys have had, but I hope you recognize gamma and delta. These are the lowercase alphabetical letters as opposed to the uppercase. Anyway. All right, let's continue on now. We'll start talking about then assembling the segments to make the beta chain of a T cell receptor. So, as I said just now, immature T cells first attempt to assemble a functional TCR beta chain gene. Okay, remember that corresponds to the case of B cells, the heavy chain. Okay. So just as the case of B cells, for the T cell receptor beta gene, we have V and D and J segments. Okay, and then also a constant component, or just like for the antibodies, we have for the light chain, we have half of it is constant, the same for all kappa or lambda light chains. The other half of it is variable. Okay, so in the same way here, we're going to pick a D from a library, pick a J from a library, that'll make a DJ pair, okay? Having made a particular DJ pair, and here it is right here. We pick this, actually what happens here is, in antibody we have a bunch of Ds, okay? Here we've, in this case, for the beta cells, we've got essentially just two Ds. We'll pick this one D, D1, okay? It's for the beta chain, so it's the diversity segment for the beta chain number 1. And match that from a small library of J segments. So we make a particular DJ pair. When we brought this D, we loop out the interval here, drawing this D to this J. Here it is, okay? And then we'll loop out the intervening, we'll pick a V box at random, okay? Loop that out. In this case, we grab the purple-blue box here and join that to the DJ. So here we are. We picked a particular, we used D1, picked a particular J, and then we brought this one, a whole bunch of V boxes here. We picked this one, and there we go. And again, we insert random nucleotides, just the way we did this with the B cell. Okay. And oh dear, it didn't work. We had a stop codon or whatever. Okay. So that didn't work. So now we have another go at it. We can now use this. Now we can pick. This didn't work. The cell senses that and says, okay, let's try it with this D2 and one of these J's. So we match up this D with randomly one of these J's. Here it is. We picked the very last J down the row here. So now we have D2 with this particular J. And now we grab any one of the B boxes which are left. This one's now gone because it got moved out and thrown away, but we have everybody to the left of the purple-blue one we use. So we're going to pick at random here. Here it shows us actually picking this one right here. I beg your pardon, picking this one. Okay, we could have picked the light blue one, but whatever. Okay, it's all random. Now we get so we have a second chance. If that doesn't work, bye-bye, sayonara, you're dead, go away. Next. Okay. And there you go. So now let's take a look at what happens with the alpha chain. Okay. So remember, the alpha chain on a T cell receptor resembles the light chain for an antibody. Heavy chains have V, D, and J. Light chains for antibodies just have V and J. And the same thing is true for the alpha chain of a T cell receptor. We just have V's and J's here. Now we've got a bunch of J's. Okay, I guess to compensate for the fact that we're not going to be using any D's. So we pick a particular V box. In this case, it's this one right here, and we picked at random one of these many J's. In this case, we picked the fourth one down here, and we made a VJ. Didn't work. Stop codon was generated. Well, now there's no more another set. We've got just this one set of V and J's, but we can go and have another go at any of the upstream V boxes again. Okay? And if by chance we picked one maybe right here, we'll have another go at it. I suppose if you picked one, you might have picked for your second go one which is way upstream. But if your second go was one that's not too far away, you still, I think, could go back, keep trying another V boxes further until you've used up all the V boxes, basically. At any rate, you have a pretty good chance of making multiple goes at it, and you have a pretty good chance of making an alpha chain. Okay. So now then, it turns out that remember the gamma delta T cell receptors? The delta chain locus, all the various of B and J boxes for the delta chain. They're in between the B boxes and the J boxes here, okay, for the alpha chain locus. Okay, so that when we succeed in matching a B box with a J box, we're going to loop out and throw away all of the possibilities for a gamma, for delta chain creation. Okay. I just mentioned that as an aside. But this is otherwise just a summary of what we have said. We will first do the beta chain locus is what we work on first. We have some goes at it here. We have two different DJ sets to pair with our B boxes. And here we create then here's constant for the beta chain 1 or the alternative constant for the beta chain 2. And there's that going to be in the bottom protein. This is the DNA coding for that protein. And then here is the VDJ that we put together successfully. So the constant domain and the variable domain, so to speak, of the T cell receptor. And then for the alpha chain, we'll have, here's the constant domain of the alpha chain. And we'll have picked, like 60 different J boxes and 70 or 80 different B boxes here will create an alpha chain. Okay. Now, the gamma delta TCR loci, the gamma chain locus is on a different chromosome altogether from the alpha and beta loci, okay? But the gamma, the delta locus, I'm going to say here, is located on the same chromosome as the alpha chain segments here. Okay. And this just emphasizes one more time that the, oh, we don't have this, never mind. So, this is a summary of what we have just said. Now, I'm not going to, there is not going to be an essay question, so to speak, where you're going to lay all this out for me. But you need to know the information because I'll ask you multiple choice or two false questions where you'll have to remember this stuff. That you start with trying to make a beta chain first and then make a light chain locus and so forth. But now I want to go through some of the details now. This looks kind of horrifying at first. It's a little bit horrifying, but it's actually pretty simple. I've mucked it all up here so I can remember myself when I'm going through my notes before the lecture. Oh yeah, that's how it goes. Okay, so anyway, here are these stem cell type precursor cells, okay? And we were able to identify them using flow cytometry and so forth. And so now we have a cell which is ready now to start cutting and joining the pieces of DNA to make alpha chain and beta chain and so forth. Now, the first thing we do is to turn on the process where we're going to cut and join segments to make the beta chain of the T cell receptor. But at the same time as we're going to work on making the beta chain of a T cell receptor, of an alpha beta T cell receptor, we have free reign on both making the alpha chain and the delta, on the gamma and delta chains. Okay? So alpha beta are 95% or whatever of all T cells. In the process, a cell, any cell coming into this can become a gamma delta or an alpha beta. Most of them are going to become alpha beta, and we're going to start working to make the beta chain of an alpha beta T cell receptor. But while we're working on that, the gamma delta guys are going at it, okay? And they don't care whether they make the gamma chain first or the delta chain first. They're just trying to do it. So here's a cell which before it successfully made a beta chain gene, made both the gamma and the delta chain genes. Okay, so boom, right out of the starting box, we got ourselves a gamma delta T cell. Okay, and this gamma delta T cell doesn't have, has not yet made a beta chain, so it's just a gamma delta T cell with not a successful beta chain. In other cases, we may have made, by the time we make a beta chain here, so we made a beta chain, but before we made a beta chain, this cell also succeeded in making a gamma chain. Okay. Here's another cell where, okay, we've made a beta chain, but before we managed to do that, we made a delta chain. Okay. And then here's a cell, we made a beta chain, but we did not succeed in making it either a gamma or a delta. Now, as soon as we make a successful beta chain gene, all DNA cutting and joining stops. Okay, everything is frozen where it was, and we start to proliferate. We want to, whenever we've made a beta chain gene, we're going to go through cell proliferation. So we're going to form clonal descendants. This cell comes, as soon as this cell is made, we make a bunch of 100 or so clonal descendants of this. As soon as this cell is made, make a bunch of clonal. We amplify that. We have got a beta chain. We're going to amplify the numbers of that. We're going to restart the process of continuing to make an alpha chain, and also restart the process of the gamma delta tries. Okay. So, in order to start proliferating, so once we've made a beta chain gene, we're going to turn on the expression of a couple of genes that make essentially a pretend alpha chain. So PT, you can think of it as standing for pretend, okay? Pretend alpha chain. It's pre-TCR alpha gene, is what that actually stands for there. But it's a pretend alpha chain gene. Okay? Just like in the case of B cells, we made a heavy chain gene, and then we express that together with a pretend light chain gene. Beta lambda to come together to make the pretend light chain, and then that B cell proliferates. Same deal here. We've made a beta chain gene. Now we turn on these genes that make a pretend kind of a pretend alpha chain, okay? And that then functions just like, and that looks like a T cell receptor. And we also turn on the expression of the CD3 signaling components as well, okay? So we've got what looks like a real T cell receptor, and it gets some kind of signal so that the cell will now proliferate as if it had seen its cognate antigen. Okay. Okay. So we have cell proliferation. After we've amplified the numbers, we resume the cutting and joining it at the level of DNA. So here's this cell that had a beta chain gene. Just the alpha, but mainly we made a beta chain gene, and it also made a gene for a gamma chain. If this cell then succeeds in making a delta chain before it makes an alpha chain, then this will become a gamma delta T cell that happens to have a functioning gene to make a TCR beta chain, but it will not make the TCR, even though it's got the gene, it won't make that protein. It's just a gamma delta T cell that has in it an assembled gene for a TCR beta chain protein. If, on the other hand, this cell succeeds in making an alpha gene before it makes the delta, it'll become then an alpha beta T cell that has in it a functional gamma chain gene for a gamma delta receptor, but it doesn't express that gamma chain gene. Here's the cell that made the beta chain gene, and then it also made a delta here. If this cell makes a gamma to match with that delta, it'll become a T cell that has a gamma delta TCR. It has, again, a functional gene for a beta chain, but it doesn't make a beta chain. If this cell here makes an alpha chain gene before it makes gamma and delta, it'll just become a gamma delta T cell receptor here. So that's how that goes. Okay. Okay. And we, by the way, these cells here, at the same time as they turn on the genes for the pre-T alpha chain and the CD3 signaling components, they also turn on the expression of the CD4 protein and the CD8 protein. So these cells that emerge out here now are double positive cells. Okay. Okay. And this just says a little bit more about this pretend alpha chain here. This just picture, I was just telling you this, the analogy, what's going on here in the T cell, it happened with the B cells. We said we made a successful heavy chain gene in a B cell, and then we expressed it together with a pretend light chain gene. This was the V3B and the lambda. These are two genes encoding a V3B protein and the lambda, so-called lambda protein there. And these resemble essentially a light chain, and they're expressed together in a heavy chain, and that makes these cells able to proliferate. And the pre-T alpha, remember we made a beta chain for the T cell receptor, and we expressed it at the surface with a pretend alpha chain. So the pretend alpha chain functions analogously to the pre-B cell receptor here with the surrogate light chain. Okay. So, so again, here we have the germline gene arrangement, the germline DNA arrangement here with all the B boxes, T and J. This is a immature double negative T cell or thymocyte. Okay, does not express CD4 or CD8. And we tried to make a beta chain, so we made a DJ pair, and we patched them and brought that together with the B box, okay, here. And so we're down to here. So now we have a B DJ thing. We have a gene that codes for a beta chain, and there's no stop codons and so forth. So we make that protein now, and then having made that protein, we're going to turn on the expression of the surrogate light chain. Okay, I'm sorry, the surrogate alpha chain. Okay, so now we've made this alpha, we made the beta gene, we turn on the genes to make a pretend alpha chain, okay? And we then also turn on the genes for the CD3 proteins which function in signal transduction, you remember. And then also we turn on the genes for CD4 and CD8. So this T cell, which made a successful beta chain, is expressing that successful beta chain on surface together with a patched up alpha chain like thing, and all the other signal machinery, and also CD4 and CD8. This is a double positive cell here. And then we'll go to work making a real alpha chain, and if we succeed with that, now we replace the pretend alpha chain with the real alpha chain, but all while we're not continuing to express CD4 and CD8 and the CD3 components here, signal transduction. Okay, so this is a double positive cell, and job one for this T cell receptor. This T cell has now made a proteins alpha and beta chain proteins, okay? But there's a lot of, we've inserted random nucleotides, so the part of the alpha and beta chains that are going to recognize the MHC peptide complex, you got to now see, well, job one is, do they bind the MHC? Remember, a peptide is going to be presented on the MHC protein, okay? And the T cell receptor is going to come up here. The T cell receptor is going to use some of its fingers to bind the MHC molecule, but other fingers to come in here and bind the peptide. And so job one is for positive selection. We're going to mainly see, does this T cell receptor tightly bind the MHC part here? We actually don't want it to bind the peptide here. Because that would be a self-peptide, you see. We don't want those. So it's going to be positively selected for binding pretty tightly to the MHC, because the finished T cell with its T cell receptor has got to have some pretty good affinity for the self-MHC. Just those two little loops. Remember, the alpha chain has 3 equivariable loops. Beta chain has 3 equivariable loops. Of those 3, in each case, 2 are binding the MHC, and only 1 of each is binding the peptide. So for practical purposes, your dependence, the tightness of your binding to the peptide is based on just the 2 loops, one from the alpha chain, one from the beta chain. And really, you need to have you need to more stabilize that binding. So that's why we think you have to have an MHC molecule, and you have to have binding on it. Or you need to have the T cell receptor has to bind both the MHC and the T cell and the foreign peptide. Okay. And your positive selection is basically just measuring that you've made a T cell receptor that will bind the MHC presenting molecule that your body makes in your particular case. Okay. So remember this for the test. Positive selection occurs when cells are double positive. That's these cells right here. They're making both CD4 and CD8. I'll have more to say about that in a moment. And here's just a little bit more about gamma delta T cells. We had talked about them before. They come out of here during embryological development. So this is I think mice looking at here, but it's presumably very similar to us in humans. Early in embryological development of the mouse, we have an early arising set of gamma delta T cells. Okay. Now the alpha beta T cells don't. We make some of them in the early embryo, but really they come up pretty much after birth mostly. But before birth, we have a set of gamma delta T cells in the embryo, and they then move to the skin of the embryo. And they'll be in the skin for the rest of that embryo to become full-grown mouse the rest of their life. Okay? So a lot of our gamma delta T cells are under our skin and so forth. And then a little bit later, just before birth, another set of gamma delta T cells arise. Okay? And these are found in the reproductive tract epithelium. And they use this one, these early ones, they use a particular V box number 5, V gamma number 5. And these ones here use a particular V1, delta box V1, and these use I have to zoom in myself to read it better. So this is the gamma V box number 5 of the many V boxes. So you use just this one V box, which has evolved evolutionarily to function for recognizing whatever the ligands for that gamma delta T cell receptor are. And this one uses the gamma V box number 6 here, not on the test. I'm just calling attention to it. And then later, you then have at the same time you're now releasing the alpha beta T cells, you now release the rest of the gamma delta T cells, and they use the gamma chain V boxes 1, 2, 4, and 7. I don't know where 3 is, but anyway, there you go. Okay. And as I said before, gamma delta T cells, they're not like alpha beta in any way. The alpha beta, I keep emphasizing, they see a small peptide on an MHC presenting molecule. The gamma delta T cell receptors, they don't have to have foreign peptides presented by MHC proteins, okay? They recognize a different type of antigen. We basically don't know shit about them. I speak, I'm sure there are highly knowledgeable, specialized investigators who would take outrage when I say we don't know shit about them, but most of us don't know anything about them. Now then, in the interest of making sure we have enough time for our review, I'm going to spare you much of the details of the next couple of pages here. This is a famous experiment, okay? And it was the experiment that we show on this page summarized and this page right here. This is of historical significance. I've already told you what it all means. This is just how we learn what I've been telling you, namely that T cell receptors, they're not like antibodies. A T cell receptor doesn't just bind some few amino acids on the surface of a whole protein, okay? It has to see a protein's got to be cut up into bits, loaded onto an MHC, and then the T cell receptor recognizes both the MHC and the peptide. In other words, T cell receptors are MHC restricted. So if you took T cells from mouse of genotype A and then MHC molecules from mouse of genotype B, the T cell receptors from mouse A cannot recognize peptides presented by the MHC from mouse B. Okay, that was basically the experiment. Okay, and that basically was the first realization that T cell receptors are fundamentally different from antibodies. They recognize their antigen only if it's presented by an MHC molecule. Okay. So that's, this is sort of mouse A and mouse B here, and this is pictures about that whole experiment. It can recognize peptides presented by the right of the MHC molecule that it grew up with, so to speak, and not, and vice versa. I'm being kind of incoherent here. I just don't want to spend 10 minutes on this, which I would do in maybe some previous lectures. This is also, this next page, page 118, it's pretty much the same. It's all part and parcel of this whole mouse A, mouse B, MHC restriction of the process here. Sort of pictures about what's going on there. Again, the whole point is T cell receptor has to see the foreign peptide on its own self MHC molecule. Okay. And now then, we come to this summary picture here. Actually, this is not a summary picture. This is about positive and negative selection here that happens in the thymus. Okay, so we have made a T cell receptor, okay, and we hope that we've generated diversity because we need to be able to recognize foreign peptides, okay? But in the process of developing the sort of huge numbers of diversity, just the way you make huge numbers of huge diversity and antibody genes to make antibodies of all possible specificities. We want to make T cell receptors of all possible specificities against whichever foreign peptide might happen to come in. Okay? But it's complicated with the T cell receptor because while we want to generate all possible diversity with respect to foreign peptides coming in, we've got to keep, we've got to maintain not all possible diversity. We need to have the self-MHC repertoire, A, B, and C, DP, DQ, DR, MHC genes, okay? And so now we're going to select. So here's a shiny new TCR. So happy to see that I did it. Look, Mom, I made myself a TCR. But I said, well, that's pretty good there. Now let's just hold on a second before we get too happy. And then sure enough, that one here. The T cell receptor, now this is the thymus, so the peptides here are self-peptides. We don't want the shiny new T cell receptor to tightly bind to a self-peptide, but we do want it to tightly bind to the MHC molecule. So here's one that does not bind to the self-MHC. Of course, it doesn't bind to the peptide either, but that's no good. Since it doesn't bind to the self-MHC, it is rejected. Okay, this did not pass positive selection. But here's another new T cell. It's made its shiny new T cell receptor, and looky here, it binds very nicely to the MHC molecule. It's just the right lock and key specificity. Now, we don't want it to bind to the self-peptide, or at most we'll let it bind very poorly. Okay, that cell, this guy will pass. Okay, move on. You get to pass. You get to graduate to the second grade. Okay. Now this guy will then get into the region of the thymus where we're now going to get to really stringent stuff here. We don't want it to bind tightly to self-peptides here. Okay? So this guy, which binds nicely to the MHC, unfortunately binds very nicely also to one of your own self-peptides. So you're done. So again, this is brutal Darwinian natural selection in action here. Okay, it's slaughter. Okay, and only the strong survive kind of thing here. In this case, the ones that are strong for MHC but weak for self. Okay, so the T cell receptors is kind of a more stringent, kind of more complicated, almost kind of qualitative test that it's got to pass. It's got to find. It's got to recognize peptides presented by a self-MHC, but here it can't bind too tightly because that means it's an autoimmune T cell receptor. Okay. And again, a picture here again, illustrating our point I keep saying over and over again. Recognition of antigen by TCRs is MHC restricted. Okay. So here's the TCR. Here would be a foreign peptide. Okay. Let's say this is a class 1 molecule here. This is a virus-infected cell, let's say, and this could be then a virus peptide. Okay. And here's the one T cell out of a million. It's already been positively and negatively selected in the sense that its TCR does bind very nicely to the MHC, the yellow MHC1 presenting molecule here. And it didn't recognize any self-peptide, so it managed to escape the thymus into circulation. But now it is the one T cell receptor out of a million that does bind tightly also to this peptide. And in that case, this is a foreign peptide, namely, this would be a virus peptide from a virus-infected cell. Right? So this T cell would kill this virus-infected cell. Now, I'm going to give you a question on the test. And I just want you, I'm going to tell you, all right, we've got, I don't know, some virus, let's call it influenza, and we've got a mouse that got infected with influenza. And so in that mouse, we will have evolved T cells, killer T cells, that recognize an influenza peptide presented by a mouse MHC, kill it very nicely. If we take those killer T cells from the infected mouse, and now do a tissue culture experiment, where we have mouse embryo, mouse fibroblasts infected with influenza virus. Those T cells taken from the influenza-infected mouse, if they see an influenza peptide on tissue culture cells, which are mouse gizzard, they're going to kill those viruses, influenza-infected mouse cells in a petri dish. But if you have that same influenza virus, and we have monkey cells, the mouse T cells we took from the infested mouse, those T cells, they will recognize, they know how to bind the influenza peptide, but they can't bind the influenza peptide if it's presented by a monkey MHC molecule, right? So the question I'm going to ask you is really simple, okay? And I'm going to do various permutations of different viruses and different cell types, but that's the very simple thing I want you to recognize. Okay? I'm telling you this now because I'm now about to launch into some other stuff where we talk about alloreactivity. I'm almost tempted to skip alloreactivity because it's just going to muddy the water for you guys, okay? You're going to, when I give you my simple question about two different viruses infecting one about the cells, and, the simple question about MHC restriction, I just told you 2 seconds ago what the question is going to be. Some of you are going to get freaked out and start thinking about alloreactivity, which I'm about to talk about here. Now, in the question, I'm going to tell you, this has nothing to do with alloreactivity, okay? But the people who are not here for this morning's lecture or this afternoon's lecture, they're going to go in that creek without a paddle, okay? And they're going to lose 10 points, okay? So congratulate yourself for being here, okay? And with that warning, in a moment now we'll get started talking about alloreactivity, but not now. Quite yet. Now, remember we said that we made a successful beta chain gene, okay, and that we made a successful alpha chain gene, okay, and now those cells expressing a new alpha beta TCR, also expressing, remember, all the CD3 signaling molecules and so forth, and also expressing both CD4 and CD8, and these are the so-called double positive precursor cells. They now go through positive selection, where we're looking to see if the T cell receptor will bind good to your particular set of MHC 1 and 2 molecules. Okay? That was positive selection. During positive selection, that's when the cell, a cell which has passed positive selection process is now defined as either a CD4 cell or a CD8 T cell for the following reason. Okay. So now here we have some of these double positive cells here. Okay. Here's one double positive cell. Here's another double positive cell. Both of these guys have made a shiny new T cell receptor. Okay? And it's going to be tested for its ability to bind to an MHC molecule. Okay? Hopefully not binding. If it binds to it, it's a cell peptide, it's going to get screwed. That's going to be the end of it. But it may find out that we have a T cell receptor that actually binds pretty well to a class 1 molecule, but not so well to a class 2 molecule. Amino acid MHC1 and MHC2, they both have the same sort of platform peptide presenting thing here, but they have different amino acid sequences on the hills and valleys all around that peptide. And so a T cell receptor might bind well to a class 1 molecule, but not very well to a class 2 molecule, or vice versa. If by chance this T cell receptor encounters a person, maybe it binds well to class 1 and class 2, but if it happens, it would be rare, but maybe it binds, it'll finally find an MHC1 molecule pretty early on, and it binds tightly to it, so good, okay? And since it's binding to a class 1 molecule, the CD8 protein will be recruited here in the usual way, and the CD4 protein will be sort of hanging in the breeze here, And this will become a CD8 T cell and stop expressing that useless CD4 protein. Okay? So the positive selection event is what also determined this double positive cell to become a single positive CD8 T cell. Here, by chance, the T cell receptor recognized an MHC2 protein well. Okay? And so its CD4 protein would be brought into play here, and the CD8 protein will be waving in the breeze, and this then will become a CD8 T cell and stop making the CD4, oh, I said that wrong, it becomes a CD4 protein and stop making CD8. Any question? Is that clear? Okay. It's certain cells here that kind of look like dendritic cells in the cartoon here, but these are these so-called cortical epithelial cells, and they mediate positive selection. So we have double positive cells coming in here, being positively selected, and then becoming either CD4 or CD8. And then as CD4 or CD8 cells, they then go through negative selection, and if they bind too tightly to a self peptide in class 1, it would be deleted or whatever. So the negative selection is essentially preventing autoimmunity. Positive selection is you have made a functional T cell receptor. Negative selection is it's got to be functional, but it can't be anti-self. Okay. So if it encounters self-antigen here, it dies and is gobbled up by macrophages here. If it doesn't encounter the self-antigen in this environment, but encounters self-antigen later on, after the thymus it becomes anergic, and then later on it will just die. So it's the same thing with these anergic T cells, like anergic B cells, they fail to thrive and they die. They're paralyzed. And this is just a summary that we're not going to worry about. And now you get to the treacherous topic of alloreactivity. Again, I don't want to get into too much detail on this. And there's something called, I think has some clinical application, sort of mixed lymphocyte reaction here. Basically, all this is saying that even though I have emphasized that a given T cell from a mouse, let's say, has got to see its cognate peptide presented by a mouse MHC protein, okay? And if it sees a monkey MHC protein, it won't react with it. Okay? Well, that's not quite true. Okay? That's 99% true. 1% it's wrong because because in a population of mouse T cells, there's going to be a few mouse T cells that will recognize a monkey MHC molecule or a peptide presented by a monkey MHC molecule. And so I'm not going to go through all the details of all this, but I'm just going to show you the picture here, which is illustrating all this. So this is the way it is supposed to work. Okay. Here's a mouse T cell, and it's recognizing a foreign peptide, like a virus peptide, presented by a mouse MHC molecule. So mouse TCR, mouse MHC. Not just mouse, but remember that there's allelic variability in MHC molecules. It's got to be a syngeneic mouse, basically, because it's got to have the right alleles of the MHC 1 and 2 molecules, kind of thing. Okay. But a foreign peptide on a self MHC, that's supposed to work. Okay. Presenting a foreign peptide on a non-self MHC doesn't work. I keep telling you that. That's the basis for the question you're going to get. It's not going to work. Okay. But here's this So it's biology. There are exceptions, okay? We could have a case where we have a mouse TCR here, and it's being presented by a monkey MHC molecule here, but it's a really strong by the two loops here on the T cell receptor that bind the peptide. Even though the other four loops, two on this one, two on this one, even though they don't bind MHC, and of course you would think have a tight enough overall interaction, because we have really tight binding to the foreign peptide, we can get away with it not being on a proper MHC molecule. That's alloreactivity. Allo means foreign, okay? This is a foreign MHC molecule, but the T cell receptor is still, this T cell is still becoming activated because of really tight binding to the foreign peptide. Okay? Or just by chance, a T cell receptor that actually really tightly binds a foreign MHC molecule. That's a rare thing, but in a population of millions of T cells for the animal, there may be the occasional one that actually will very tightly bind a foreign MHC molecule. So that's alloreactivity. Do not get confused by alloreactivity. I'm telling you this for the 5th time now. The question I'm going to give you on the test has nothing to do with alloreactivity. My question on the test is going to be just the simple, basic self-MHC, foreign peptide, bingo. Non-self-MHC, foreign peptide, nothing. Okay? Normally, it's T cell receptors are MHC restricted. Here we're talking about rare cases where T cell receptors are not MHC restricted. Okay? Or over MHC restricted or something like that. And the main reason your book goes into this is for a kind of a complicated reasoning that addresses the question that professional immunologists have been, so this is sort of pure science type of question here. I'm just going to go through it with you relatively briefly. If you don't quite follow me, that's because I don't quite follow the logic myself. See? But the conclusions have some interest, okay, and I'll just talk to you about it. So remember I said we have 3 genes to make an MHC1 molecule, A, B, and C. And each of those can bind about 10,000 different peptides. So the A MHC can present 10,000, B a different 10,000. So about 30,000 peptides total we can recognize if we have the A, B, and C gene. And of course we have mom and dad versions, so we've got actually 6 genes that make class 1 molecules. So 60,000 peptides can be bound to the MHC1. I'm just keeping it simpler by talking about MHC1. The same thing always true for the class 2 molecules as well, of course. So if 6 genes is good because we can present 60,000 peptides out of the universe of all possible peptide sequences. Actually, 60,000 peptides may be impressive, but it wasn't good enough for the Native Americans, remember, when Columbus sailed the ocean blue, right? Brought all the nasties to some. It's not a complete capability to bind critical peptides. So why don't we have 6 genes from mom and 6 genes of class 1 from dad, so a total of maybe 12 possible MHC1 molecules representing now 120,000 possible foreign peptides. And the argument that's made here, the reason that doesn't happen is because then you start getting too much autoreactivity. Then you start making, if you can bind not just 60,000 but 120,000 peptides, it's going to be more and more challenging to make sure that you're not recognizing the self-peptides among that 100,000, okay? So if you have just a few number of MHC genes, you don't make enough functional T cells, basically. If you have just the right number of MHC genes here, you could have optimal number of population of T cells, even considering negative selection and so forth. But as you start getting even more MHC genes, you fewer and fewer cells in circulation because they're just being slaughtered by the negative selection. They're just too many MHC genes is presenting too many cell peptides, and it overall affects the actual functional number of T cells you have. So that's the argument that's made here. And I follow much of the argument, but there are aspects of the argument I'm saying the book says. So it's not, you can see why, if this is really than this, and I'm going, no, I don't see that. Why? So, but whatever. So, your book now that includes with, remember when we talked about the details of B cell differentiation? We said different kinds of leukemias come from different kinds of B cells, different kinds of mutations in different B cells in different stages in the process of development. And the same thing is true, certain kinds of leukemias and lymphomas arise in cells at different stages in the differentiation process. Okay, here. So we get acute lymphoblastic leukemias, B cells, adult T cell leukemias of B cells. I'm not going to go through it all. It's not on the test. Okay. Same thing was true with B cell, but you didn't have to memorize what cancers come from what stages of development. And also in the case, last time you could do Southern blotting here and you can follow the treatment of a particular T cell leukemia or lymphoma by Southern blots, which are based on your code recognizing restriction enzyme fragments from germline regular 39% mutation rates. And so you can pick up these rearranged pieces here. The graduate students, you had to know that in the case of B cells for the previous test. For the second test, I'm not going to repeat myself. This will not be on the second test for undergraduates or graduate students. The idea is basically the same thing as what we talked about for B cells. So we'll take a pretty nice little break here. And we have an hour or so to do an exam review. So we have finished our lecture topic for today, and we'll come back after the break and go through what we need to know for the test.